Alright, so welcome to your video covering the leftovers from the July 25th lecture for BISC 202. So when we left off, we're talking about other kinds of operons. So we talked about two operons, the ribose and the lactose operon, for catabolizing different sugars as energy and a carbon source. And we're starting to talk about biosynthetic pathways and how, for prokaryotes anyways, the enzymes tend to be organized into operons such that, let's say you needed to make tryptophan, an amino acid, you would transcribe and translate the tryptophan operon, which would help you to make all of the enzymes needed to make tryptophan, right? And it will make it actually, it'll transcribe each gene in the order in which the enzymes that they produce are used in the biosynthetic pathway. So the first structural gene transcribed and translated will make the first enzyme in the biosynthetic pathway, the second one makes the second enzyme, and so forth. And I left you by asking you to think about how that is actually more efficient than having them just in some random order. And it kind of makes sense because remember that in prokaryotes, translation can start while transcription is still happening. So if you imagine that your RNA polymerase is coming along and transcribing each of these structural genes in order, then a ribosome can start translating the mRNA as soon as it's being produced, and in doing so will make the first glutamine, the first enzyme for this biosynthetic pathway. So you can basically start these first reactions first. Then next thing you know, the ribosome has now translated the second enzyme that's needed, and you get the second enzyme needed to get this reaction going, and so on and so forth, right? So evolution has placed these structural genes in the same order in which the enzymes are used in the biosynthetic pathway, because that actually kicks off each reaction that's needed in the order from beginning to end. Right? So you get more efficient production of tryptophan from the very get-go when you start transcribing and translating this operon. Okay? Something to think back to is that when we started talking about biosynthetic pathways and different mutations that make a prototroph into an auxotroph, so these auxotrophs might be trip minus or met minus, we're talking about the mutation of an actual operon. Right? So whereas before we kind of just called them met and trip genes that are either wild type or mutant, right? What we're actually mutating are operons instead of single genes. Okay, so there's a clarification of what was actually mutated in these auxotrophs in our previous lectures. All right. Anyways, moving on then. So we're going to look at one last operon that is quite different from your lact and arabinose operons in that we're not taking a sugar and breaking it down. We're actually going to be synthesizing something that the cell needs. All right. So the regulation is going to be a little bit different from these lac and arabinose operons because instead of making enzymes that are going to catabolize or digest sugars, we're going to be synthesizing something that's needed. Right. So if your role is to take a sugar and break it down, then when the sugar's around, that sugar is going to induce expression of these operons. Whereas on the other hand, if you have an operon that's used to synthesize something like tryptophan, it should only be turned on when there's not enough of tryptophan in the cell. So in other words, if there's lots of tryptophan around, that should actually come back as a negative feedback loop and turn off this operon, so we're not making more tryptophan than we need. All right, so that's something to keep in mind, is that for these ones, you add sugar, it turns it on so you can digest that sugar. Here, you make it when there's not enough tryptophan, and when there's enough tryptophan around, that should negatively feedback and shut this off. Okay, so presence of tryptophan means shut this off, we don't need it anymore. Okay, so let's take a close look at how a tryptophan operon is regulated. All right, now there are two different mechanisms for regulating this operon. The first one looks similar to what we've been doing so far. The second one's quite different. So let's look at the first one first. So as we said before, when the cell has enough tryptophan around, so there's a bunch of these red tryptophan amino acids floating around the cell, right? Then that causes the TRP repressor protein to bind to this excess tryptophan, and together they can now bind to the operator sequence of the tryptophan operon. Okay, so when the operator is blocked by the tryptophan repressor and TRP complex, then we see negative regulation. So this is quite similar to the sugar operons in that we have negative regulation. So basically, when a protein binds to the DNA at the operator sequence, we're going to block transcription, shut down transcription. But the difference is that with the lac operon, adding the sugar makes the protein come off. This is the other way around. When there's tryptophan around and it binds to the repressor protein, this is what actually makes it bind onto the DNA even more strongly. Okay, so when tryptophan binds to the allosteric site here of the repressor protein, it increases its affinity for DNA at the operator sequence instead of decreasing it. Okay, so lactose makes the repressor come off, tryptophan makes this repressor go on the DNA. Okay, so this makes sense because you want to block transcription when there's plenty of tryptophan around, and if there's no more tryptophan, then this repressor should come off so you can make some more. So in terms of terminology, thinking back to the lac repressor protein, that system of controlling the lac operon, right, is called an inducible system because you can induce expression of the lactose operon when you add lactose as an effector molecule. All right. So considering that the lac repressor protein is an example of an inducible system because you can add lactose and induce transcription by getting that repressor off of the operator, then TRP repressor being a slightly different system, what would you call that? Right. So maybe pause the video and think about what would you call the type of control that the TRP repressor is an example of. So it's not an inducible system because it behaves differently from the lac repressor. Right, so pause the video and think about it. All right, welcome back. So the answer to that question is it a repressible system? Okay, so in terms of looking at this TRP repressor protein, when you add the effector molecule, when you add the thing that's bound to the allosteric site, right, the site that doesn't bind to DNA, the result is that you repress gene expression further. All right, so lactose induces lac operon expression by binding to that repressor protein, whereas tryptophan represses tryp operon expression when it binds to the TRP repressor protein. Okay, so a lot of terminology there, but if you make a little table or something like that, you can try to organize differences between positive and negative regulation, inducible versus repressible, systems of control, and so forth. Okay. All right. So here's another question. So what if there's a mutation in this TRPR gene here, such that when it's transcribed and translated to make the TRP repressor, right, the resulting protein that's made by this mutant TRPR gene can no longer bind to tryptophan amino acids. Okay, so if that's the case, what do you think will happen? Pause the video and think about what the correct answer is and how you'd explain it. Okay, so pause the video now. All right, welcome back. So the answer is constitutive expression, right? So if this repressor protein is messed up because the TRPR gene is mutated, such that the repressor protein can no longer bind to DNA at all, right? Then it's no longer able to block expression of the tryptophan operon, and therefore you'll see constitutive expression means that you'll see that this operon is turned on and you're making lots and lots of these enzymes all the time, whether there's tryptophan already there or not. Okay? So if you have questions, you can ask your tutors or TAs or myself later on. All right. So that was one mechanism, is kind of a repressor protein binding to the operator sequence, blocking transcription from happening. It's all pretty familiar based on your lactose and arabinose operons as well. The only difference being that the effector molecule makes it bind more strongly rather than less. It adds the repressor on rather than taking it off. So the second mechanism is quite a bit kind of more wonky, and that's called attenuation. So attenuation meaning that you're going to stop transcription early. All right, so basically, normally if you want to make your your tryptophan producing enzymes, right, you would transcribe everything from here, the transcription start site, all the way through all the structural genes. Whereas if you don't want to make those enzymes, you can start transcription but end it early before you even get to the structural genes, so that you're not wasting as much time and energy making enzymes that you don't currently need. All right, so attenuation means ending transcription early. Okay, so this ending of transcription happens at the attenuator, right, which is the sequence here. And ending of this transcription happens again when there's lots of tryptophan available, right? So remember, if there's lots of tryptophan available, we don't need these enzymes to make more tryptophan if we already have lots. Okay, so to understand how an attenuator works in terms of how do we end transcription early, it's important to remember the mechanism we talked about earlier in terms of how transcription is normally terminated at the end of a gene or operon. Okay, so this is a quick review of the intrinsic mechanism for ending transcription normally. Okay, so generally speaking, like transcription, the elongation transcription happens as the RNA polymerase moves along the DNA template strand and transcribes that onto mRNA, onto RNA. Okay, now transcription is terminated when you get to the termination sequence at the end of the gene, right? And that termination sequence is usually around 40 base pairs long, and it ends transcription in a specific way. Okay, so in that termination sequence, there's two very important regions that you'll find there. There's a GC-rich region, followed by a bunch of A nucleotides, right? So you see here in the template strand, CGCCCG, right? So a bunch of G's and C's, followed by a whole bunch of A's. Okay, so how does that cause transcription to end? So the way it does this is RNA polymerase comes and transcribes those important sequences, that termination sequence, and once that sequence has been transcribed onto RNA, the RNA itself starts to fold in on itself. Okay, so that GC-rich region becomes this becomes a GC-rich region in the mRNA, and that base pairs with itself to form this hairpin loop. Okay, this hairpin stem loop here. So these triple bonds between each GC pairing forms this really stable and strong loop here. Okay, and then after that, RNA polymerase also adds a bunch of U nucleotides to match the bunch of A's in the DNA template strand. Okay, so those AU pairs, since there's only two hydrogen bonds between each of those are very unstable. So what RNA polymerase normally has to do is backtrack from these bunch of A nucleotides to try to find a GC pair to stabilize itself before it continues transcription. But unfortunately, these GC pairings here have blocked RNA polymerase, so it can't go back. It can't find a GC pairing. It's just stuck right here among those A's and U's that are really unstable. So since RNA polymerase can't go backwards, can't find a GC pairing because it's blocked by the hairpin loop, and it can't go forward because all the AUs are super unstable, it basically gives up and everything breaks apart and transcription is terminated. Okay? So how does this relate to your attenuation of transcription in your trip operons? So so right here, notice that we have the promoter, right, which is going to be where RNA polymerase binds in order to do transcription, right? The operator sequence where it can be blocked by the repressor protein, but there's this leader sequence, right, that's between the promoter and operator and your structural genes, right? This is not translated into any important enzymes or useful proteins, right? It's just a bunch of DNA that's found in between the regulatory regions and your structural genes. Okay, so within this long leader sequence, right, there's an attenuator, which is another termination sequence. So normally you have a termination sequence at the end of the operon or at the end of the gene. This one has it early here within the leader sequence. Okay, so when this attenuator gets transcribed, right, you see the same result as before, the same as we saw in the termination sequence down here, in that the mRNA itself will fold up into a hairpin loop, and there's a whole bunch of UA base pairs, right, that can end up ending transcription because it's unstable and the RNA polymerase can't go backwards. All right, so what happens then? So when there's lots of tryptophan around, what happens is that this hairpin loop in the attenuator will stay closed, right? So if this hairpin loop stays closed, there's a bunch of UAs here, RNA polymerase is going to get stuck, can't go forward, can't go backward, and we end up stopping transcription early. Okay, so transcription ends early. It's only if there's really low tryptophan that something will open up this hairpin loop, right? Will cause this hairpin loop to open up so that RNA polymerase can actually go backwards, find a GC pairing, right? And continue to move forward and transcribe the rest of the gene. Okay, so the key is either there's lots of tryptophan around in which case the loop stays closed and termination happens early at the attenuator, or something comes along and opens up the hairpin loop when there's not enough tryptophan around, and therefore allows transcription to finish across the structural genes. Right, so a challenge question for you to think about is this. So the hairpin loop will end transcription early, right? It'll make the resulting RNA short so that it doesn't contain structural genes as long as this loop stays closed, right? So something is able to open up this loop structure when there is not enough tryptophan around. So a question for you to think about is, what in the world, what structure inside the cell, right, can actually force this hairpin loop to open up, okay, so that RNA polymerase can back up and then continue forward and transcribe the rest of the structural genes, okay? So think about it. Which of these four structures can somehow come along and open up this hairpin loop? Right? So it's a difficult question, but I want you to think about it because it really helps you understand how everything fits together. So a very big hint is that this process only occurs in prokaryotes. Okay, so what kind of interaction with this RNA can only happen in prokaryotes and not in eukaryotes? Right, so pause the video and think about it, see if you can choose the right answer. Okay, welcome back. So the correct answer is ribosome. Right, so let's go through these possibilities. So RNA polymerase. Well, RNA polymerase is already doing what it can to transcribe the gene onto RNA, but it's stuck because it can't go forward through these UAs without being able to go back to the hairpin loop. So RNA polymerase can't open this loop. If it could, then it would never terminate transcription. Okay, so it's still stuck downstream of the hairpin loop with these U's and A's. It's not going to open up this loop on its own. The ribosome makes sense because it's only in prokaryotes that the ribosomes and the DNA is not separated by a nuclear envelope, right? So as transcription happens, ribosomes are moving along this 5' end of the mRNA. It's starting to translate already. So if it moves far enough into this hairpin loop, the ribosome can basically open up this hairpin loop by coming into for translation purposes. Okay, so that's something that in prokaryotes that can happen. Just to go over the other remaining wrong answers, so repressor protein is not going to help open up the RNA because it binds the DNA instead, right? So wrong type of target. And also the repressor protein represses gene expression, so it's not what we're looking for when we're trying to increase full gene expression. Also tryptophan amino acids themselves, having them around shouldn't be opening up the hairpin loop, because if we have lots of tryptophan around, why do we want more complete transcription anyways? Okay, so let's look at how this works. So how does a ribosome open up this loop to allow transcription to finish? All right, so let's look at what happens to the leader sequence. So when the leader sequence gets transcribed into RNA, right, so we have the DNA here opened up, we're transcribing it onto RNA, and this RNA has these 4 yellow sequences here, right, these 4 yellow regions that can base pair with whatever regions right beside it. Okay, so if there's no ribosome in the way, if nothing else is going on, this is what the mRNA ends up looking like once it's transcribed, right? So there's regions 1, 2, 3, and 4, right? The best way to base pair such that every region has something to base pair tightly with is to put region 1 and 2 together and put region 3 and 4 together. Okay, so region 3 and 4 is that hairpin loop that's right beside a bunch of U nucleotides, right? So it's with 3 and 4, and then the 3 and 4 making a loop, and then a bunch of U's here matching the A's on the template strand. That's where termination would happen early if this stays closed. Okay, so when tryptophan is low, what happens is the ribosome is going to try to translate this mRNA. So the ribosome is coming along from the 5' end of this mRNA, translating, adding amino acids, building the protein, and so forth. When it gets to region 1 here, these 2 orange boxes represent codons coding for tryptophan, right? And so the ribosome is going to wait here until tryptophan can be brought in to continue doing translation. So if there's not enough tryptophan around, then the ribosome is just sitting here waiting for more tryptophan carrying tRNAs to come in, right, and get stuck there, right, until tryptophan can come along. So if the ribosome is stuck here for a while, then that's getting in the way of 1 and 2 binding together, so it breaks up this loop here. So region 2 is going to be like, okay, I got to bind to something now. I can't bind to 1, the ribosome's in the way. So region 2 binds to region 3 instead. So notice that this loop is made by region 2 and 3. Now region 4 is wide open, right? So now RNA polymerase can continue its job. RNA polymerase can now back up into region 4, find its GC pair, stabilize, and continue transcription across all the structural genes, right? So because the ribosome got stuck in region 1, because it's waiting for these tryptophan codons, it needs 2 of them, it doesn't have enough tryptophan to do that for now, right? Then that blocks region 1 from binding to region 2. So region 2 goes ahead and binds region 3, making a loop between 2 and 3, unlike 1 and 2. Okay, so 2 and 3 gets joined together, and then region 4 opens up so that RNA polymerase can now back up onto region 4, find its GC pair, and then continue transcribing the rest of the structural genes. So then what happens when the concentration of tryptophan is actually high and we don't need to make any more tryptophan? Well, when the concentration of tryptophan is high, then the ribosome doesn't get stuck right here with these tryptophan these codons that code for tryptophan, so it doesn't get stuck in region 1. Instead, it basically continues to translate this leader sequence of the mRNA until it gets to a stop codon, which is around the end of region 1. So as it stops here further down, it not only blocks region 1 from binding anything, it also blocks region 2 from binding to region 3. So since region 2 and region 3 are no longer bound together, then region 3 is free to bind to region 4. So again, this loop between 3 and 4 stays bound together, and therefore RNA polymerase can no longer back up into region 4 in order to find a GC pair in order to continue transcription. Alright, so again, when there's lots of tryptophan around, the ribosome goes further along the mRNA, thus interrupting both regions 1 and 2, leaving 3 and 4 free to pair with each other, so RNA polymerase cannot continue transcription beyond this attenuation sequence, and therefore RNA polymerase has to come off and stop transcribing early before you even get to structural genes. Alright? So those are the different situations. So when left alone, 1 and 2 bind together, 3 and 4 bind together, transcription would end, right? If there's not enough tryptophan, the ribosome stays here in region 1, 2 and 3 bind together, and that actually leaves 4 hanging out there, allowing RNA polymerase to back up and go forward. Whereas if there's plenty of tryptophan around, then it goes further, opens up 1 and 2, leaves 3 and 4 to bind together, allowing RNA polymerase to continue on. Okay, so a quick question to help you review this. So what happens when tryptophan is low? Just look at these answers. So pause the video and choose your answer. Alright, welcome back. So yes, the answer is all of the above. Right? So all of these things happen when the concentration of tryptophan is low. So just remember that both the repressor protein here, right, and the attenuation mechanism, which happens inside this leader sequence and with the attenuator, both of them are active at the same time. Both of them help us to block or they help us to block the transcription and translation of these structural genes, either by blocking it at the operator sequence or by ending transcription early. All right. So overall, how do these two regulatory mechanisms affect the expression of the tryptophan operon? So when concentration of tryptophan is low, 2 things happen. So without tryptophan to bind to this repressor protein, it doesn't bind well to the operator sequence, and therefore the tryptophan repressor is detached and floating around on its own. So with the removal of the tryp repressor, then you get around 70 times more transcription. So way more transcription happening when you're not blocking it with the repressor. In terms of attenuation, it's not as strong an effect, but it is kind of multiplicative in that you get about 10 times more transcription when you have low concentration of tryptophan, and therefore you're ending transcription less often at the attenuator, like the termination sequence of the attenuator here. All right. So overall, right, depending on whether there's lots of tryptophan present or not enough tryptophan present, you can see about a 700 times difference in how much this operon gets expressed, how much it gets transcribed and translated. when we compare like very low concentration of tryptophan situation compared to very high concentration of tryptophan condition. All right. So if you do have questions, do take some time next week to ask your tutors and your TAs and myself, since we do only have one more week before tutoring ends. Okay, one last thing. So in terms of attenuation, right, another question you can ask yourself is, okay, does this happen in other amino acid synthesis pathways as well, other biosynthetic pathways? And the answer is yes. You'll notice that each of these different operons which are used to synthesize a different amino acid, they all have a sequence that requires some of the amino acid that they would synthesize, right? But notice that the number of codons that code for that amino acid, so the number of amino acids, number of codons that code phenylalanine in the operon that synthesizes phenylalanine, there's way more, right? You need 7 of them in order to get past this leader sequence, compared to the 2 we needed for tryptophan. And we needed 7 of these histidine amino acids to be brought in order to get past this part of the leader sequence, right? So to review, so for the CHIP operon, how do we kind of stop attenuation in order to make more tryptophan? Well, as long as we don't have enough tryptophan around, if we have less than 2 tryptophans, we're not going to be able to continue translation beyond this point. So the ribosome stays here, 3 and 4 stuck together, 2 and 3 are stuck together, 4 is open to allow RNA polymerase to back up and continue transcribing and translating all of the structural genes, and therefore we'll be able to make more tryptophan until there's enough that we can start having attenuation again. Basically, if we don't have enough tryptophan, we want the ribosome to end up being stuck here because there's not enough tryptophan to do translation. All right, and that allows us to make more tryptophan. Now, for phenylalanine operon, notice that you need to have less than 7 phenylalanine amino acids that are already pre-attached tRNA available in order for the ribosome to get stuck and therefore get past the attenuation sequence and continue making the structural genes to synthesize more phenylalanine. Right? So basically, kind of same setup for tryptophan, phenylalanine, and histidine, right? But the number of codons coding for that amino acid is different, 2 versus 7 versus 7. All right? So the reason for this is that the amount of each amino acid that you need to have inside of the cell in order to efficiently make the polypeptides and proteins you need to make is different, right? So based on how often these codons appear in the leader sequence, kind of tells you what relative concentration of these amino acids we need in the cell, right? So if we want to have a higher concentration of phenylalanine in the cell, then basically we can allow the transcription to complete itself even when there is more than 3 times as much phenylalanine around compared to tryptophan, right? So you can have 3 times as much phenylalanine than tryptophan and still not have enough to finish translation past these phenylalanine codons so that the ribosome gets stuck at the right point to allow transcription to finish so that attenuation does not happen. Same with histidine as well. All right, so the number of codons coding for that amino acid helps us kind of control how high a concentration of the amino acid can be present in the cell while still synthesizing enzymes to make more of that amino acid. Alright, so this stuff is quite complicated. It takes a bit of thinking to figure out, so I do suggest you review this lecture again with the lecture recordings, but also to watch other videos and read other textbooks to see different perspectives and see it presented in different ways so that you know it beyond just the words and examples I've shown you here. Alright, thank you so much. So you have now have enough information to do your master genetics practice and quizzes. And we'll just have one last topic to cover next week to finish off the semester. All right, thank you very much. Have a great weekend, and I will see you all soon.